So the Biochemical Society and Portland Press are pleased to welcome you to this webinar, which is part of our Biochemistry Focus webinar series. Topics in the series include different research areas in the molecular biosciences, as well as practical sessions to support career development. Each webinar will give you an opportunity to ask questions via text, and we welcome suggestions for future topics and speakers to feature in our webinar series. Please see the website for more details. I'm Dr. Hannah Dugdale. I'm a lecturer in molecular and cellular physiology in the School of Sport, Exercise and Health Sciences at Loughborough University. I specialize in skeletal muscle adaptation within the context of disease and performance, and I'm very pleased to be able to chair this session today. Within the Biochemical Society, I'm a committee member of both Research Area 5, so biological systems, and the Clinical Translation Research Theme Panel. These positions have allowed me to meet a wonderfully diverse group of people, both from very similar and completely different research areas, and I've also utilised the Biochemical Society training to gain new skills, helping me with both my teaching and research areas, such as R programming. So today's session is part of our dedicated early career researcher programme of webinars. And we are delighted to provide this opportunity today for our speakers to share their work within the molecular biosciences community. Our speakers are Hiran, James, Silvia, and Francesco. Today's webinar is titled Clinical Metabolism, Exploring Therapeutics and Treatments, and we will hear from these 4 early career researchers who will share their current work within this field. Now, before I hand over to our first speaker, I'd like to mention that the questions will be asked at the end of the webinar, but please send in your questions during their talks. If you do have a question, please type it in the question box as shown on the screen now, and make sure to state who your question is for. We will try and answer as many of these questions as possible at the end. So our first invited speaker today is Hiran Prag from the University of Cambridge. Hiran is a research associate with Professor Thomas Krieg. After studying pharmacy at the University of Nottingham, he pursued a PhD in mitochondrial biochemistry, supervised by Professor Mike Murphy at the University of Cambridge, where he is today. Hiran is exploring mitochondrial dysfunction in myocardial infarction and aiming to develop novel therapeutics. Today, Hiran will discuss his work on targeting mitochondrial succinate metabolism to treat ischemic reperfusion injury. And without further ado, I'll hand over to Hiran to let you know about his research.
Thank you very much for the introduction, Hannah, and thanks for the opportunity to present some of my work here at this Biochemical Society webinar. So, as was mentioned by Hannah, my main focus of my research has been in myocardial infarction or heart attacks. And this occurs when there's a blockage in the coronary blood supply, often, for example, through atherosclerosis. And so underlying tissue is starved of blood flow, oxygen, and nutrients. Clinically, it's really important that the blood vessel is reopened. Normally, the patient has to go to hospital to get this done. And in order to get the blood flow going again and to salvage the tissue and the function of the heart. But paradoxically, when this blood vessel is reopened, this causes even more damage by this process or phenomenon called ischemia reperfusion injury. So historically, it was thought to be just this random chaotic event because oxygen is coming back into the tissue and electrons leaking onto the oxygen, causing reactive oxygen species production and oxidative stress leading to this damage. But more recently, it's been found that everyone's favorite organelle, mitochondria, are a key to this process. And as you can see here in green is the mitochondria within a cardiomyocyte. And it was found about 10 years ago that the Krebs cycle metabolite succinate is a key driver of ischemia reperfusion injury. So to study IR injury within the context of a heart attack, the model that we use mainly in the lab is this left anterior descending, or LED, coronary artery ligation in the mouse. So you can wrap a suture around the LED coronary artery and press it against it to occlude the blood vessel. And when you do that, you create a nice ischemic lesion, ischemic area within the left ventricle. It's really nice, a well-defined model. And to mimic what happens in the hospital on reperfusion, you can just open up this suture and reperfuse the blood vessels, and you have this infarct within the left ventricle. And so if you take a mouse heart and make it ischemic for 30 minutes, and then analyze the levels of succinate by mass spectrometry imaging, what you can see in this slice of the mouse heart is that there's a huge accumulation of succinate selectively within the ischemic region of the heart. And the rest of the heart is pretty much at normoxic levels. So the healthy heart, the succinate levels remain quite low. So this phenomenon of succinate accumulation within ischemia is not solely limited to the mouse, but also we see the same phenomenon occurring in the pig heart, but also in the human heart as well. And so this suggests that succinate accumulation is a conserved part of ischemic heart tissue, or ischemia in general. So on reperfusion, this succinate is rapidly returns to normoxic levels. So as you can see here, most of the heart looks pretty much the levels of succinate are the same. And that's because some of the succinate leaves and enters the circulation, but a large proportion of the succinate is rapidly oxidized by succinate dehydrogenase within mitochondria. So the consequence of this succinate oxidation by succinate dehydrogenase is that it can drive this process of reverse electron transport at mitochondrial complex 1, which generates these free radicals, superoxide, which can prime subsequent cell death and injury in cell death and damage in IR injury. So I'll just walk you through this quickly. On reperfusion, oxygen is now no longer limited. Oxygen has come in, so the electron transport chain can start to work as normal. And so a consequence of this, you get proton pumping, and you generate a large proton gradient across the mitochondrial inner membrane. In these early reperfusion conditions, ATP synthesis is quite limited because during ischemia, the adenine nucleotide pool is heavily degraded, and so there's a lack of ADP for real ATP synthesis, and so the delta P isn't being used up, and so you remain having a large proton gradient there. Also, succinate oxidation by succinate dehydrogenase maintains a reduced coenzyme Q pool, and so the consequence of this is that electrons from the CoQ pool travel backwards up through complex 1 onto the FMN and onto oxygen subsequently, generating superoxide, which is rapidly dismutated to hydrogen peroxide, and together with other factors initiate this damage. So the question that we wondered is whether we could actually target succinate metabolism to be therapeutic and to actually prevent ischemia reperfusion injury. So what we used is this really classical inhibitor of SDH, which is malonate, which acts in a competitive manner. As you can see, it's quite structurally similar to succinate. It's relatively well tolerated as a single dose, and you can give it at relatively high levels because it's metabolized and excreted quite efficiently by the body. And so when we give malonate into these animals at the clinically relevant point of reperfusion, it's administered, just a note, it's administered as disodium malonate. So if you see DSM, that's all it means. It's just the disodium salt of malonate that's being given. What we see is that in the saline normal treated animals and the at-risk tissue, which is the tissue that was previously ischemic and then was reperfused, the levels of succinate return pretty much to their non-risk healthy tissue levels. But when malonate was infused, this seemed to prevent succinate from being oxidized by succinate dehydrogenase. And so the succinate levels seem to be higher than they were, or than they are with the control treated animals. And so this suggested that the malonate is getting into the mitochondria. It's inhibiting SDH and slowing down succinate oxidation. A consequence of this is that by treating the animals with malonate and slowing down succinate oxidation, we seem to reduce the formation of reactive oxygen species within this at-risk region of the heart. So we measured this using the mitochondria-targeted probe, MitoB, which was developed in Mike Murphy's lab a while ago now. Essentially, MitoB accumulates around a few hundred to 1,000-fold selectively within the mitochondria and can react with hydrogen peroxide to form MitoP. And so you have this ratiometric probe that you can measure by mass spectrometry. And we can see here that malonate leads to a lower MitoP-B ratio, so there's less hydrogen peroxide produced within the malonate-treated animals. As a consequence of slowing succinate oxidation and lowering ROS production, we also found that the malonate dose-dependently reduced the infarct size, so the amount of damage that was in the left ventricle of the heart. And so we could get really nice, robust cardioprotection using malonate at the clinically relevant point. This was really intriguing, and it seemed as though we could target succinate metabolism using malonate, and that could be a really good thing. But there's also a really interesting phenomenon we found with malonate in that it seems to be able to get into ischemic tissue preferentially compared to normoxic heart tissue. So in this experiment, we've got a mouse heart where, well, different mouse hearts that have been held ischemic for different times and then reperfused with malonate. And then the malonate levels subsequently measured by mass spectrometry. And what you see is that with the hearts that have been exposed to a greater period of ischemia have more malonate in them on reperfusion. So they've been reperfused with the same concentration of malonate, but more malonate seems to get in when the heart's exposed to ischemia. So we're really curious about what is causing this increase. So what potentially could be happening to allow malonate to preferentially enter the ischemic tissue on reperfusion. And when we thought about the metabolic changes that occur within ischemic tissue during ischemia, there's a large accumulation of lactate, and the pH drops significantly within ischemic tissue. And that was the first avenue that we went down because of some previous data that we had that it could well be an interesting mechanism of acid playing a role in malonate preferentially getting into ischemic tissue on reperfusion. So when we infused malonate into hearts under a normal pH, very little actually got into the heart tissue. But when we used a low pH buffer, this seemed to really drive malonate uptake into the heart. And so we had a theory that this could be due to malonate being protonated into its monocarboxylate form at low pH. And so by decreasing the pH, we drive the protonation and increase the proportion of the malonate in its monocarboxylate form. This is interesting because then malonate could potentially act as a substrate for SLC16A1, or the monocarboxylate transporter 1, which is a canonical lactate transporter. And so it transports the monocarboxylate lactate together with a proton across the plasma membrane. And so we wondered whether MCT1 could drive malonate uptake across the plasma membrane of cardiomyocytes, and then malonate can subsequently enter mitochondria and have its effect. And so when we repeated this same experiment using low pH and malonate uptake into the heart, we found that using a potent and selective MCT1 inhibitor, this completely abrogated this pH-dependent effect of malonate. And so it suggested that malanate's effects, or malanate's preferential effect, could well be to do with the local pH changes that occur within the ischemic tissue. But that was all well and good. That was just using a low pH buffer and infusing malanate into hearts. How about when we actually did some ischemia and reperfusion? So one of the things that we wanted to check was actually, is this an MCT1 dependent phenomenon that's occurring in here? So when we did do the same with ischemia, so we held the hearts ischemic for a set period of time and then reperfused with malanate, we saw a nice increase in the amount of malanate that was in the reperfused tissue. But when we used an MCT1 inhibitor and co-administered this with the malanate, it seemed to, and co-administered this with the malanate, it completely again blocked this ischemia selective uptake into the heart on reperfusion. And so it suggested that this ischemia selective uptake of malanate into the heart tissue was dependent on an MCT1 mechanism. We could also leverage this in the opposite way by forcing malanate into its monocarboxylate form using a low pH or acidic malanate formulation. And so we could give a low dose that was not protected in its neutral form, but when we used it as an acid form and drove malanate uptake into the cardiomyocytes, we could actually achieve robust cardioprotection using a low dose, which is great for minimizing off-target effects. So overall, what we've been able to show here is that targeting succinate metabolism may well be a legitimate strategy to prevent ischemia reperfusion injury, especially within the heart in this context. And by acidifying malanate, there's the option to drive its increased uptake into cardiomyocytes as well. So we're taking this forward a bit more now. We've shown that there's some efficacy within the space of the heart, also longer term protection, and also for other organs such as ischemic stroke, where we're testing out the utility of malanate. So it's an exciting time, I think, and watch this space for future stuff. Okay, I'm just going to end with a couple of acknowledgements. I'd just like to thank Thomas Krieg and Mike Murphy for their labs, and the people in the lab for all their help and support. Richard Hartley at Glasgow, one of our chemical collaborators, has been essential, and Dunya Aksentijevich in Queen Mary. Also, Zhelong Zhu's lab in Tianjin, and AstraZeneca, Richard Goodwin and Andreas Danhorn for mass spectrometry imaging. And that's it. I'd just like to end there. Thank you very much.
Fantastic, thank you for that, Hiran. Very informative presentation. I'm sure there'll be a number of questions associated with that we can address at the end of the session. So next we have our second invited speaker. This is James Lee from the University of Sheffield. James is a final year PhD student in Professor Heather Mortiboy's lab studying mitochondria in ALS. Previously, James graduated from the University of Bath with an MRes in Biosciences and from the University of Exeter with a BSc in Medical Sciences. Today, James will present his work titled C9orf72 repeat expansion mutation reduces mitochondrial function and mitophagy in neurons. So, James, if you want to take it away.
Yeah, thanks very much for the introduction, and thank you to the Biochemical Society for the invitation to present at this webinar. As just mentioned, I'm going to be presenting some of the work from my PhD, which I'm just wrapping up over the next few weeks, looking at C9orf72, the most common ALS causing mutation in Europe, and its impact on mitochondrial health in neurons. So a bit of background on ALS, first of all. It's the most common form of motor neuron disease, which is an adult onset neurodegenerative disease that's characterized by progressive loss of upper and lower motor neurons, shown on the image on the left. Survival is typically about 3 to 5 years, so it's a really devastating disease. Symptoms typically include a progressively developing muscle weakness resulting in paralysis before the end stages of the disease. In terms of causes of the disease, we see about 10% of cases have a family history, and looking primarily at those patients, we found about 30 mutations at the moment, which are associated with an increased risk or affect the progression of the disease. The other 90% of cases refer to as sporadic ALS. And at the moment, there's very limited treatments. There's only one approved treatment in Europe. In the US, they're approving slightly more, so they're up to about 4 approved treatments. All of these, unfortunately, have very limited efficacy. So there's a lot of drive at the moment in the community of ALS researchers to try and develop new treatments for the patients and their families. So as I said, I study C9orf72, which I'm just going to call C9 now because the key name's a bit of a mouthful. So as I said, it's the most common mutation in European cohorts, accounting for about 40% of familial ALS cases, and it's also seen relatively frequently in sporadic ALS cases as well, making up about 10% of those. The mutation is a hexanucleotide repeat expansion in intron 1. So there's this G4C2 repeating region in the gene, in that intron. Normally repeats a few times in most people, but in people considered to carry this mutation, it will repeat usually several hundred to several thousand times. This has several impacts on the protein and several toxic gain of function effects as well. So it reduces transcription and hence translation, so reduces the amount of functional C9 protein that's produced in cells carrying this mutation. This protein has several roles in synaptic transport and the initiation of autophagy. Also, when you get the translation of it and the mRNA is produced with these repeat expansion sections, these can fold up and form a number of complex structures which sequester RNA binding proteins, which can be quite toxic to the cell just on its own. And also you can get the production of dipeptide repeat proteins when you get RAN translation, so translation of these repeat sections without start codon. So 5 different DPRs or dipeptide repeat proteins can be reduced, each of them contributing different amounts and different degrees to cellular toxicity. So as I said, I study mitochondria in ALS. The reason being, we found that metabolic dysfunction appears to be a hallmark of ALS pathology. So some of the key findings in ALS patients described in this image on the left from a review several years ago, showing several indications of hypermetabolism in ALS patients. And when we look at more cellular models or even in animal models at the cellular level, we see a range of different areas in which mitochondrial health, mitochondria being some of the key energy producing organelles in the cell, we see various areas in which these are affected. So I've summarized just 4 of them here, probably the 4 most commonly studied. So these are impacts on the respiratory train complex, so complexes 1 through 5 of their assembly and activity. This can have a knock-on impact on things like mitochondrial membrane potential and mitochondrial ROS production. Disruption to mitochondrial ER contact sites, a shift towards more mitochondrial fission, and although it's not been hugely well characterized until it's becoming increasingly more of interest to ALS researchers, there's some indications from certain protein and gene expression studies suggesting mitophagy is decreased in ALS. So some of the aims from my work was to try and characterize this mitochondrial phenotype in neurons from C9 ALS patient cells. And we hypothesized, based on some of the previous work that's been done in fibroblasts in C9 ALS, that there'll be a shift towards more fragmentation of mitochondria. There'll be reductions in mitochondrial function, and mitochondrial turnover or mitophagy will also be reduced. So in order to look at this, we generated neurons from directly reprogrammed iNPCs. So typically, the common method for generating neurons from patients is to take fibroblasts from the patients, reprogram them with these Yamanaka factors into iPSCs, so induced pluripotent stem cells, turn these into neural progenitor cells or NPCs, and then turn these into neurons. We've skipped one of those stages, so we reprogram the fibroblasts directly into iNPCs. And one of the reasons we've done this is we found that these retain a lot of the aging-associated epigenetic changes. which would typically be lost if you reprogram into iPSCs directly. So I've taken these, differentiated them into neurons over an 18-day protocol, and put them through a range of different live and fixed imaging assays and a seahorse bioanalyzer. And for reference, these are the control and patient cell lines that I've used for this study. So first of all, I took some cells. I labeled the mitochondria with mitotracker green and imaged them on the opera phenix that I showed briefly on the slide before, put them through the software that we've got that analyzes those images, and outputted, or segmented mitochondria and outputted the parameters we've got for mitochondrial size, so area and length, and shape, so a roundness score and width-length ratio, which is slightly different, but essentially measuring kind of the same thing. And we don't see any significant differences there in the neurons that I looked at. Just to make sure, I also outputted the data for each individual mitochondria for area and length, so I could visualize the distribution of how these mitochondria are distributed across the different sizes. And essentially, these lines kind of overlap, they're essentially overlapping, suggesting that the populations are almost identical between controls and patients. And to give reference, this is about 6 million mitochondria in each condition, so control and patient mitochondria. So a really huge population that I've surveyed here. Next, I did some mitochondrial functional assays. So the first, in that same assay from before, I also labeled those mitochondria with TMRM, so a dye which accumulates the mitochondria in a membrane potential dependent manner. So the intensity of this dye in the mitochondria indicates how high their membrane potential is. However, we see a slight decrease in our patient mitochondrial membrane potential, about a 20% decrease in the intensity of this dye, indicating these mitochondria are slightly less healthy than their counterpart controls. In order to try and confirm some of these findings and look at oxygen consumption, I used the seahorse bioanalyzer that I mentioned before, and put the cells through one of the standard protocols on that machine, which is a mitochondrial stress test, which produces a graph similar to the one you see in the middle here. So there's various different parameters we can get from this, but essentially we don't see any significant differences here. And I've plotted for an example the basal respiration, which we see at the start before we administer any toxins over the first 20 minutes. And we don't see any significant differences in oxygen consumption between controls and patients there. So next, I've done more of an in-depth characterization of mitophagy, because it's not really been that well explored in C9 ALS before. So to start off, first of all, I took some cells without any induction conditions for mitophagy and fixed them and labeled the mitochondria in green on these images and the autophagosomes in magenta, or the pink color, with the marker LC3. So based on what I've tried to highlight with these arrows here, the co-localization of one of these autophagosomes with the mitochondrial network, seeing how many of these mitochondria co-localize with autophagosomes compared to the total number of mitochondria, we see a significant decrease, about a 60% drop in the amount of mitochondria being co-localized with autophagosomes in our patients relative to controls. So I mentioned that C9 is involved in autophagy initiation, largely via an interaction with ULK1. So to double check if comparing some of the stuff to the previous literature, I also outputted the amount of LC3 puncta, the density of LC3 puncta, so how many per micrometer squared of cell area. And we see about a 60% drop in that as well, suggesting this may be driving this mitophagy deficit in our cells. Just to make sure, we've done some other characterizations. So looking at the co-localization of mitochondria with lysosomes, we don't see a significant drop here, suggesting that delivery of mitochondria to these lysosomes doesn't appear to be impacted in these neurons. I've also looked at induced mitophagy via administering oligomyosin and antimyosin A and imaging the cells for a 90-minute time period. And by comparing the induced condition to basal condition, the amount of mitophagy occurring, to look at the fold increase in mitophagy, we see about a 50% increase in mitophagy with this mitochondrial insult of these toxins. And that's the same across controls and our patients, suggesting that their ability to increase PINK1 Park-independent mitophagy doesn't appear to be impacted as a result of this, the C9 mutation. One of the other key mitophagy initiating pathways is this protein BNIP3, which is inserted into the mitochondrial membrane to highlight the mitochondria for degradation. So we looked at this under the co-localization of this marker with mitochondria under basal conditions and induction via deferoprone, a chronic 24-hour treatment with a high dose of deferoprone. And we found under the basal conditions and these induced conditions, there was no issues in the C9 ALS patients with their ability to co-localize and recruit BNIP3 to the mitochondria and trigger mitophagy that way. So yeah, that suggests that basal and induced mitophagy don't appear to be affected by issues with BNIP3 dependent and PINK1 Park independent mitophagy, two of the key mitophagy initiating pathways. So this led us to look back at this autophagy initiating stuff that I mentioned before, looking at ULK1, which I said has a known interaction with C9. So one of the ways we try to get around this mitophagy and autophagosome producing deficit is using compounds which have been shown previously to increase ULK1 activity. So nilotinib is a drug that's been developed for non-small cell lung cancer and has been shown to increase autophagy in those cells. BL918 has been shown to increase ULK1 activity. It's a known tool compound for increasing its activity. So with that compound, with its highest dose in a 24-hour treatment, in our control neurons, we did get close to a 40 to 50% increase in mitophagy and a slightly smaller increase in autophagosome production. We do see some slight increases in our patients, but not enough to rescue them back to the levels of our healthy control cells. So that's pretty much it for me. So just to wrap up, in the C9 ALS neurons that I've looked at, we see reductions in mitochondrial membrane potential, indicating an impaired function of the mitochondria in these neurons. The cells also appear to have a reduced ability to clear these damaged neurons via mitophagy, but this does appear to be independent of PINK1 Parkin pathways or the BNIP3 pathway for highlighting mitochondria for clearance. And increasing ULK1 activity does not appear to be able to rescue this mitophagy deficit. So some of the future work I'm wrapping up at the moment is investigating whether ULK1 recruitment is impaired to the mitochondria, and also testing whether we can rescue this effect with C9 or F72 gene therapy, so reintroducing the healthy gene. So just a few quick acknowledgements. Thank you to my supervisors, Heather, Scott, and Professor Pam Shaw. One of our collaborators, Professor Laura Ferrioli, who developed the technique for generating the directly reprogrammed IMPCs. Members from all three labs, and to my funders, the MNDA and Patel. So yeah, thanks again for listening, and I look forward to your questions later on.
Thanks for that, James. That was brilliant. It's really nice images in there and a very comprehensive data set that you've got there. So let's move on to our third speaker before we get to those questions at the end. And remember to put your questions in the chat if you had some questions for James in there as well. So our third speaker is Silvia Radenkovic from the Mayo Clinic. Silvia is a postdoctoral fellow at the Department of Clinical Genomics. Her research focuses on using tracer metabolomics to investigate underlying metabolic adaptation and possible therapeutic options for inborn metabolic disorders, or IMDs, specifically congenital disorders of glycosylation. Today, Silvia will present her work titled Investigating Therapeutic Mechanisms for Aldose Reduction Inhibition in PMM2-CDG. So without further ado, Silvia, if you'd like to take the stage.
Yes, hi, thanks for having me. Let me just change my screen because, can you see the presentation now?
Yes. Yeah, that's it.
Okay, awesome. So today I'm going to talk a little bit about one of my PhD projects, and this is about investigating therapeutic mechanisms of aldose reductase inhibition in PMM2-CDG. So first of all, I would like to say I have no conflicts of interest to declare. And I'm not sure how many of you know about congenital disorders of glycosylation. They are very rare genetic disorders, but you might have heard about glycosylation, which is a complex biochemical process that produces chains of sugars that we call glycans. And here you can see one of the materials that we actually use in the clinics for the patient education, and it shows a simplified version of glycosylation. And here you have a cell membrane, and with the different sugars, so we have mannose, fucose, galactose. They enter the cell and they build nucleotide sugars. These can then be important in ER and Golgi, where the sugar chains or glycans are formed and attached to the proteins. So when you have a fully functional glycan and the protein together, these are exported to the membrane surface and they can perform their functions. Glycosylation is important for protein integrity, cell-to-cell adhesion, immunity, a lot of different things. And because it's so complex, it is not unusual that we have more than 160 CDGs described up to date. So any of the problem in this huge pathway can lead to a congenital disorder of glycosylation. They were first described in Leuven in Belgium in 1980s, and this is actually where I did my PhD, started my PhD, and that's how I got interested in them. However, even though after 40 years, we don't have any therapy for most of them, and diagnosis and treatment remains a really big challenge. There's a lack of biomarker and treatment options. So it's a very sad story, and we're trying to find new ways how to treat these patients. The first CDG that was described, and actually we have the most patients belonging to the CDG, involves phosphomannose 2, and this is an enzyme that converts mannose 6-phosphate to mannose 1-phosphate. So if you have a defective PMM2 enzyme, you cannot make mannose 1-phosphate and therefore GDP mannose. GDP mannose is one of the nucleotide sugars which is involved in glycosylation. So if you don't have enough of it, you cannot make full glycans. So patients with this disorder have pathogenic variants in the gene coding for phosphomutase 2, and majority of individuals with this disorder have nervous system involvement. However, other organs are also affected, like liver, heart, coagulation, and so on. So these patients are usually seen by a multidisciplinary team, and they require really complex care because multiple organs are affected and they can be affected to a different extent. As I mentioned, there is no treatment for majority of these disorders, and PMM2 is not any different. However, recently we teamed up with a company which was actually founded by one of the patient's parents, and they found that one of the drugs that is actually used in diabetes is a potential drug candidate for PMM2 CDG. So what does drug do? Aparistat is actually an aldose reductase inhibitor, and it inhibits glucose to sorbitol formation. So it was a little bit of confusing why this would come in a hit for PMM2 CDG, because it's not really related and it's not really in the same pathway. So we decided to check in our patients whether they have increased polio levels, and the easiest thing to do that was to check their urine, and we actually had 50 patients, which is quite a lot for a genetic disorder, and we had seen that urine sorbitol was actually increased in more than 85% of our patients, while urine mannitol was increased in about less than 50%. So there was really something going on with the polio pathway. So we wanted to see whether it correlated with the severity of the patients. And we found that the more urine sorbitol the patients had, the more severe their neuropathy was. And this is here presented as a neuropathy score. And the same went with liver. So the liver was more affected in the patients that had a higher urine sorbitol concentration. So it seemed that really sorbitol was involved in the pathophysiology of this disorder. it was not enough to just look into the urine. We wanted to know where these polyols were coming from, and we looked into the patient fibroblasts. As you heard before from James, these are easily obtainable from the patients, and we can do different experiments with them. Unfortunately, they're not tissue specific. As for instance, if we want to look into the brain, it doesn't tell us much about brain metabolism, but it's a good way to check whether your hypothesis might be true. So we checked for different polyols in the fibroblasts, and we found that indeed sorbitol, mannitol, and the whole polyol pool was increased intracellularly as well. And so then we wonder what would be the mechanism of eparestat in PMM2-CDG. We actually took these fibroblasts and we measured PMM2 enzymatic activity, and we found that it increases after we added the eparestat. So this was very interesting, because why would eparestat increase enzyme activity for the enzyme that was not in the same pathway? And because it's increasing the enzyme activity, we also checked for a glycosylation status, and we found that the fibroblasts that we treated with eparestat also had increased glycosylation. And here this was done by glycoproteomics. So we saw that multiple different peptides were improving after we added eparestat. So to further investigate the mechanism, we checked for metabolites linked to the polyol metabolism first, and we found that in the fibroblasts, we could see different changes. And again, as I said, we didn't really know how these are related, because sorbitol is not in the same pathway as the glycosylation. So we measured sorbitol, mannitol, and other polyols in the fibroblasts, and you can see them here. So we showed that eparestat does decrease the polyol pools, which is known, and this was good. However, we then wanted to see whether the metabolites related to PMM2 were increased, and we also found that if we add eparestat, mannose-6-phosphate, mannose-1-phosphate, and GDP-mannose, which are all in the PMM2 pathway, were increasing. And most importantly, we found that the activator of PMM2 glucose 1,6-bisphosphate was increasing after we added eparestat. So this provided us a link to why eparestat was able to increase the PMM2 enzymatic activity. So by increasing glucose 1,6-bisphosphate pools, it was able to activate PMM2 and redirect glucose back towards glycosylation. So we did a lot more experiments with zebrafish. We used tracer metabolomics as well. We knocked out aldose reductase in these fibroblasts, and we really showed that the glucose flux was altered in PMM2. And if you block aldose reductase, you could really see that glucose was going back to glycosylation. This was all great, but we really wanted to see whether we can see any changes in the patients. So we had 3 patients that were on eparestat therapy, and one of them at the time of when we were wrapping up this publication was there for 24 months. And we could really see that their urine sorbitol and urine mannitol were decreasing the longer they were on therapy. So this was also a sign for us that urine sorbitol and mannitol might be good biomarkers to track the therapy as well. Apart from that, we saw improvement in ataxia scores. So this is tracking neuropathy, liver enzymes, and coagulation. This is again not a real trial, because we only had single INDs for these patients, which meant that we mostly looked at the safety and efficacy of this drug over a period of time. And right now we have a clinical trial, phase 3, going on with this drug, and we hope that it's going to come to the clinics very soon. So fingers crossed. So here we see a patient, the first patient that was trialed on eparestat. She has been on it for almost 4 years. She's doing great. And it is her mother that actually founded a company and made this research happen. And I just wanted to conclude that we saw that PMM2C patients have increased urine sorbitol, and the deficiency of PMM2 results in a change in glucose flux and increase in intracellular sorbitol. And if you use eparestat or other aldose reductase inhibitors to target sorbitol production, you can actually push the glucose back towards glycosylation and therefore improve it. For those of you that are more interested in the clinical aspect of this, we showed that Pauristat decreases urine sorbitol and results in clinical improvement in the 3 individuals that we treated for 1 to 2 years. And finally, urine sorbitol is a biomarker for PMM2-CTG, and it can be used to predict severity and monitor therapy. Now, I will just leave this at the last slide, and I just wanted to say that we are moving away from the fibroblast research, and this is mostly what I have been doing during my postdoc, and we are actually interested in looking specifically into the brain and how it's affected in PMM2-CTG, because more than 90% of the patients have central nervous system involvement, and we have certain doubts that a Pauristat can pass the blood-brain barrier, and we want to test whether the metabolism is different in the brain. So to test this, we actually develop brain organoids. So we take the iPSCs from the patients, so we can take fibroblasts, we reprogram them into iPSCs, sort of like James is doing with the neurons, but instead of doing neurons, we actually make brain organoids. So in you get little spheroids, and then you can grow them in the lab, and you can test them as well with the same omics methods that we used. And currently this paper is in revision, and we actually got really interesting results, which are maybe contradictory to what we see in the fibroblasts, and it really shows us that it's important to look into the tissue-specific models in order to understand the phenotype of the disorder better. And I just want to thank all my team. It's a very big team. We're at Mayo Clinic, also in Belgium, and we are supported by the patient organization as well as NIH. And if you have any questions, please let me know. And thank you again for having me here.
Brilliant. Thank you for that, Silvia.
Problem.
I'm not sure many early career researchers get the full gamut of cells all the way into clinic, so a little bit jealous there, but fantastic presentation.
Yeah,
we'll hopefully talk about it some more at the end. That was brilliant. Thank you. So if we move on to just our final invited speaker for today, Francesco Capriglia. I believe I'm pronouncing it correctly, but if I'm not, please correct me when you start. From the University of Sheffield. Francesco is a postdoctoral research associate, and his research involves reprogramming patient-derived fibroblasts into induced neural progenitor cells harboring the LRRK2 G2019S mutation and subsequently differentiating them into neurons. Today, Francesco will present his work entitled Autophagy and Mitophagy, Investigations in LRRK2 Parkinson's patient fibroblast-derived dopaminergic neurons. I hope I got that right, and it's over to you, Francesco.
Thank you, Anna. Thank you for giving me the opportunity to present to this webinar. So I'm going to start with a brief introduction of Parkinson's, just to explain the disease. So Parkinson's is characterized by a loss of dopaminergic neurons in the substantia nigra. And the typical symptoms are tremors, slow movement, stiffness, depression. And most of the time, the typical pathological finding is aggregation of alpha-synuclein in cells that they form Lewy bodies. So when we move to the molecular disorders, we have different things like alpha-syn accumulation, mitochondrial dysfunction, lysosomal dysfunction, and also the defects in autophagy and specifically also mitophagy. So as you may already know from James' presentation, my lab is very based on mitochondrial studies. So all during my presentation will be based on studying the Parkinson's, focusing on what's happened in terms of mitochondria. And about Parkinson's disease, we can have, of course, sporadic Parkinson's or familiar Parkinson's. And for this presentation, actually during my project, I've based on familiar Parkinson's. So I have both work on LRRK2 mutation and alpha-synuclein mutation. And for this presentation, I will just focus on the LRRK2 mutation. So before going ahead, I will just give a small introduction on what is LRRK2. So LRRK2 stands for leucine-rich repeat kinase 2. It's actually a very big protein. It's a 286 kilodalton protein expressed at low levels through the body. So this protein is composed by an enzymatic domain, which is composed by a ROCK domain and a core, and also the proper kinase domain. Also, we have a C-terminal domain and an N-terminal domain. So for what concerns Parkinson's, we have several point mutations that they occur in the enzymatic domain. And for this study, I'm just focused on the G2019S point mutation, which is also one of the most studied and common mutations in Parkinson's. And the effect of this point mutation is actually a gain of function mutation. So due to the mutation, what happened to LRRK2 is just like an increased kinase activity, that leads to increased lysosome formation, membrane protein accumulation, and also an increased protein-protein interaction. So the model system we use, I'm using actually during the project, is just like induced dopaminergic neurons. So James already explained the part going from patient-derived cells, so going from skin biopsies to reprogramming induced in iMPCs. So what I'm actually doing during my project is just like differentiating the iMPCs into dopaminergic neurons, which is a protocol that takes 27 days. And on these cells, the workflow I'm actually doing to performing the experiment is just moving two iMPCs, and then we plate the iMPCs into a 6-well plate. And then on the first stage of the differentiation, it's happened into the 6-well plate. Then when we move to the last 2 weeks of the differentiation, we replate the cells into a 96-well plate, so they are ready to be treated when the differentiation is finished. And then at the end of the differentiation, we stain an image. So we both staining with antibodies, or we also can do live experiments. So we stain with dyes that can stain in just the mitochondria or the lysosome, and then we image the stain plate on the Opera Phoenix. And this is like an example of the data, and then we analyze the data with the Harmony software. So this is an example of how we perform the mitophagy. So this is actually a fixed plate stained with a couple of antibody, LC3 that stain the autophagosome, because it is a protein on the membrane of the autophagosome, and TOM20, which stain the mitochondria. So this is how it looks during the analysis. So we select the LC3 puncta, and also we segment the mitochondria, and we select the mitochondria. So with this double staining, and of course we also use the OH for the nuclei, with this staining we are able to count actually the LC3 puncta per cell, or the mitochondria per cell, and also looking at the co-localization between the LC3 and the mitochondria, we have an idea about the mitophagy, because it means that when they co-localize, we are in a phase in which the mitochondria and the LC3 they are in the same area. So we are in the autolysosome stage of the mitophagy. So for this presentation, I'm going to start with a couple of slides about the LRRK2 phenotype, and then I'm going to move to one of the compounds which we are using, which is a LRRK2 inhibitors, because the goal is, because of the G2019S mutation is actually a gain of function, so there is an increased function of the LRRK2 activity. What we are doing is actually treat these cells with LRRK2 inhibitors to see if we can rescue the Parkinson's phenotype in this kind of cells. So here on the left is just like the mitochondria per cell in our control cell line versus the LRRK2 cells line, which, as I said before, they are they harbor the point mutation G2019S. So as you can see in this graph, there is an increased number of mitochondria in the mutated cells, and here on the here on the right is the mitophagy. So the colocalization that we have between LC3 lysosome and mitochondria, and we normalize it for the number of mitochondria, so we kind of get the percentage of mitochondria that are into the mitophagy. So for this graph, actually, when you see n equal 20 or n equal 18, it's actually every dot is a single differentiation, and every dot is the average of 3 different replicates. And also for our analysis, we actually measure the number of mitochondria in more or less 1,000 cells. And going forward, in this slide, we also measure the lysosomal area per cell and the number of lysosomes per cell, and on the right, the lysosomal sites. So as we can see on the left, there is an increased lysosomal area per cell in the LRRK2 cells compared to the control, as well as an increased lysosomal number per cell compared to the control. But actually the size of the lysosome is the same. So it's just like a matter of number of lysosomes rather than their size. And now, as I said before, once we have an idea of what this mutation is doing on our cells and on the parameters we are actually testing, we move to test different LRRK2 inhibitor. So for this presentation, I'm only showing this GSK25, which is actually an inhibitor that reduced the LRRK2 kinase activity, which is actually ideal because, as I said before, in this mutation, one of the effects is actually the increased kinase activity. So the graph on the left, we can see how, as I showed before, vehicle versus LRRK2 versus control, we have an increased lysosome per cell, and when we are going to treat with the LRRK2 inhibitor, we have a decreased number of lysosome per cell in a dose-dependent way. So for this experiment, I tested 4 different concentration of the inhibitor, so from 0.1 micromolar to 4 micromolar. And the same effect we have when we look into the lysosomal area per cell. So again, the number of the area of the lysosome is higher in the LRRK2 cells, and when we treat with the LRRK2 inhibitors, we have a dose-dependent decrease. And actually, this is very nice how with the treatment at 4 micromolar, the area per cell just come back almost to the level of the control one. Then for the total lysosome area per spot, we have no difference between patient and control, but when we treat with the inhibitors, we have an increase both on the control and on the patient cell line. Now, for concerning these parameters specifically, because of the resolution we have in the Opera machine, we actually are not able to get the single lysosome area. So this increase may be not due to like an actual increase in terms of size, but may also be that the lysosomes are more clustered together. So they just, the machine just like the analysis software just count the single one more like lysosome together as a single one. And after this, I just perform the an analysis about the mitophagy, and for this compound, there is actually no effect at all in terms of mitochondrial number. So as you can see on the graph on the left, there is no effect on the number of the mitochondria, but when we go on the mitophagy index, there is an increased mitophagy, which is actually mostly happen on the control cell line. And when we move to the the Parkinson cell line, there is an increase, but it's actually just a trend. So this is n equal 3, but I'm pretty sure that if I do another couple, this will be statistical, because it just like is more or less 20% more. And yeah, that's it. Let's to summarize. As I said, I worked on patient-derived cells with and then we differentiate in neurons, and the LRRK2 patient cell line, they have an increase mitochondria per cell, lysosome per cell, as well as lysosome area per cell, and a decreased mitophagy. When we treat these cells with LRRK2 inhibitors, we observe a reduced lysosome per cell in both control and LRRK2 patient cell line, a reduced lysosome area per cell, and an increased lysosomal sites. But we have no effect on the mitochondrial number, but we see an effect on the mitophagy index. And ongoing studies, what we are doing at the moment is also the data I've shown you now in terms of GSK compound treatment. They are all fixed. We are also doing target engagement to actually evaluate that the inhibitor treatment as actually an inhibitor of the kinase activity. So we are doing both. We are assessing this both by Western blot and also PLA, and also we are performing several live assay, like to measure the mitochondrial membrane potential, the mitochondrial ROS, and also live autophagy and mitophagy. And also we are testing another couple of LRRK2 inhibitors. One of is MLI2, which is one of the most used in literature, and another one which is GSK, another GSK1. And yeah, I'd like to finish just the acknowledgement, and I'm gonna say thank you to all the Mortiboy's lab, and especially with the other Mortiboy, which is my PI, Tia Parker, which used to be one of my technicians, and Rhiannon, who was a master student that now is a PhD student. Thank you.
Brilliant, thank you, Francesco. And I'm sure we should have some questions for all of our candidates now. Definitely some interesting data here that should start us off on some interesting conversations. If you are going to ask a question, please try to remember to put who that question is for within the chat as well. So we have one question at the moment. Not entirely sure who it's for, but I think maybe it's for James. Definitely not a silly question, as the questioner has suggested. So I think this one's for James, and it's a suggestion that because you've induced, you have an autophagy that's associated with your data there, could fasting help within your particular area, within your cohort?
Yeah, it's a good question. So fasting has been looked at in some ALS models. I don't think it's been looked at particularly in C9 ALS. We have looked at some autophagy and mitophagy inducers, which induce pathways which are kind of downstream of the fasting response, such as things like AMPK, and we've not found any significant impacts. So it's not something I've looked at yet, but potentially a possible future experiment.
That's great, thank you. Okay, so while we're waiting for a few questions to come in, I have one for, oh, bear with me. I have one for Hiran in terms of your dosing. So you're undergoing your ischemic and then reperfusion, and you're immediately dosing with your Malinate. So within clinic, how do you see that taking place? So would it be within a surgical procedure, or is there an issue associated with how long you wait to dose with that Malinate, or would you do it as a like preventative mechanism, for example?
Yeah, so that's a really good question. So in terms of what we do with the mouse model, we can't quite mimic what happens in a human, but it's pretty much similar, because you have a catheter that's in there. You can infuse drugs just before you pull out the clot. So pretty much at that time would be ideal. So then you have a rush of melanate that can bathe the ischemic tissue before it's about to be reperfused. So that's kind of how we would see it in terms of timing for administration.
Okay, that's brilliant. Thank you. And do you see the prolonged period of that ischemic insult being affecting whether your melanate works or not to a greater degree? So you obviously get a greater influx, but does it affect the actual outcome downstream of your phenotype following that dosing?
So in terms of the long-term protection, so that's something that we're currently working on. It looks quite good at this point in time. That's all I can say for now.
Brilliant, thank you. So I guess, we've got another question. So this is associated with making the INPCs. So this could be for Francesco or James, I believe. So how long does making INPCs or iPSCs take, and is it considered an ideal amount of time?
Do you want to go, James?
Yeah, I can have a go. So yeah, I put on the slide that it was about 18 days. Realistically, some can take slightly longer. But yeah, typically, we do have a published paper from 2014 where the full protocol is described that I can send to anyone who needs it. Yeah, for iPSCs, I believe it's a relatively similar time frame. It's just the main benefit of the INPCs would just be retaining the age-associated epigenetic changes. It does have some limits in terms of you're then only able to turn those into neurons and glia, a few types of cells, as opposed to the much larger selection of cells you get from iPSCs. But yeah, potentially the phenotypes we get can be generated in a shorter time frame from the cells that we're differentiating from them. I think that's the main benefit we've seen from our lab.
Great, fantastic, thank you. So we have another question here from Hiran. So mindful that melanate is a competitive inhibitor, how do you explain that it has had an effect even at a low dose? What is the actual concentration?
I don't know what the actual concentration is off the top of my head, but we have we've essentially measured. So a lot of the time we quantify melanate and succinate together in the tissues. So we've done all those, and we've done the maths to actually understand what. So in the higher dose, there is melanate's given at quite a considerable concentration. So the lower dose is within the realms of the concentrations that could inhibit succinate at the concentration that it's at during ischemia and early reperfusion. So yeah, but that's a really good question, because obviously we can't go too low. If we go too low, then we don't see a protective effect, as one would expect.
Great, thank you. So I have one for Silvia. So I found it really interesting the way that you kind of went towards this in terms of choosing the drug. It obviously comes from like an outside opinion, and you weren't originally sure how it fits within your pathway analysis there. So I guess my question is, do you think it's coming from diabetes originally is where it was dosed? So do you think it could work with some of your other congenital disorders? Or potentially, do you think it has a role in, say, something like performance or aging? Do you think it has like more far-reaching uses?
So for our disorders, the majority of the patients that we are seeing are pediatric, and we cannot treat some of them. So this drug was actually approved in Japan in adults. So this is also why we had safety and had 3 pediatric individuals, because we needed to know whether the doses are tolerable and so on. So far, we see the urine sorbitol in PMM2 deficiency. However, we have some other disorders where we also see urine sorbitol increase, but it's not consistent across all the patients. So we are still not sure how in those disorders the increased urine sorbitol is affected. So we really, some of them have mannose metabolism problems, so then it's more related to PMM2, but there are some of them that are not related at all. So probably something about the sugar processing and how the cell is able to redirect glucose. But I don't know if it's far-reaching. I think aparestat is still very untargeted. So we really want to have something that would treat PMN2 at core. And I think in the end, we would have to go to gene therapy. We just want to have something in the meantime, because to get to gene therapy for all these disorders is going to take some time, and I'm not sure if that's feasible at all. We really need to have something on the market for these patients because they have nothing.
Thank you. While we're waiting to see if there's any more questions, one more just for Francesco. I guess what we're interested in here is kind of translation, and I appreciate that I've worked on a particular muscle disorder before where we're looking at very different disease, almost diseases within the same cohort, because you've got different mutations, different phenotypes. But I'm wondering, have you seen similar phenotypes in other mutated versions of your disorder? And do you think there could be a universal kind of mitochondrial-based therapy associated with that?
Well, saying just like a universal thing, it would be ideal, but I don't think so. In terms of phenotype, yes, we have seen for what we are looking for, the same phenotype in other genotype. Like, for example, as I'm working on the alpha-syn line, and they also show an increased mitochondrial number, lysosomal number, defect in mitophagy, autophagy. So most likely one of these drugs, if there is like a drug which is very mitochondrial based, it will be good for different genotype as well as sporadic one, because one of the future of good, let's say good, it's not good, but one of the characteristic also of the LRRK2 mutation is as a very phenotype similar to the sporadic one. So all the GWAS studies, they correlate very much the sporadic and the genotype. In terms of like treating in this moment, in terms of mitochondrial base, there are the classic mitochondrial treatment like vitamin B, coenzyme Q10, that for sure are useful, but in this moment it's not like that you give that and you find a cure. But still a lot to use.
Yeah, it's wishful thinking, I guess.
Yeah.
We've got one last question from the chat, again for Francesco. So this individual is curious to know more about the LC3 puncture, how it was counted from the images, and also if and how LC3I and LC3II were distinguished. Thank you.
For the antibody we used, it just like count both 1 and 2. So of course, if you run a Western, you'll see the double band. For the counting, as with the software we have, we segment the cells, we segment the image region of the cells, and then we get the spots, it's actually the punta, and then we can normalize the punta for the number of cells or even for the image region, depends. For the punta is, and if your cells are almost the same image region, you can just go for the number of cells. For all the normalization we used to do in our lab for the mitochondria, we prefer normalize for image region.
Fantastic, thank you. So with that, unfortunately, I've got more questions, but we need to bring the session to an end. Thank you all for attending, and thank you very much to our speakers, Hiran, James, Silvia, and Francesco. You can continue this conversation online by following the Biochem SOC and PP Publishing on Twitter, or X as it's known now. We welcome suggestions for future topics and speakers, as I said at the beginning, and this is in particular to feature in this Biochemistry Focus webinar series. If you have any ideas for a webinar in 2024, we invite you to submit a proposal for an upcoming webinar, and you can find more information about these webinars and propose your webinar and watch previous recordings as well as this one again if you'd like to at www.biochemistry.org. All of our upcoming webinars are listed on our website as well. And if you've missed any of the more than 70 webinars that we have up there already, then feel free to watch them again here. Please visit them on the website, as I said, or even on our YouTube channel. The details will be on the screen for you now. So finally, I just want to highlight that it's more important than ever to stay connected and engaged with your fellow molecular bioscientists, and you can do this by joining the Biochemical Society's community of researchers and specialists and take advantage of key benefits, including discounted registration fees for society courses and meetings, exclusive access to a wide range of grants and bursaries, personal online access to 2 of their journals, and more. And again, please visit the website for this information. Within the Biochemical Society, as I said at the beginning, I'm a committee member for both Research Area 5, so that's Biological Systems, and Clinical and Translational Research Theme Panel. And these have helped me to meet many people and listen to such great research within a diverse area and field that is biochemistry. I've also utilized these courses, which you can find again on the website, including certain courses such as Learning R. So once again, I just want to thank all of our speakers and everyone for attending, and for those individuals that were able to provide us with some interesting questions. And again, feel free to talk to these individuals through our Twitter feed that's available. Thank you all, and goodbye.